Record. All right. So chapter 9 begins this whole process of how information, genetic information, is passed on from one generation to the next here. So today we are looking at the basic introduction of this whole process, and that is really cellular reproduction, how cells reproduce. Now, today we are going to be basically looking at somatic cells, okay? Today we are looking at somatic cells, and I'll just write it on here because I have more space. Somatic. Ooh, I don't like that. That's too light for me. That's too light. Somatic. Ah, there we go. Somatic. And somatic means body. Okay. Somatic means body. Soma is what means body. Okay. So pertaining to the body. We are looking at this, somatic cells. Okay. This is anything that's not gonads. Anything not gonads. Okay. So that's what we're looking at here. So we're going to be looking at mitosis. We're going to be looking at the chromosome structure because it's different from a eukaryotic and a prokaryotic. We'll talk about a eukaryotic first, and then we'll look at a prokaryotic at the very end. It's very simple and basic, prokaryote, as you would imagine. Going to look at this thing called the cell cycle. We're going to look at what happens when you can't control the cell cycle, and that's cancer, the C word. No one likes to say it out loud. We're going to look at ways that you can help prevent it and what constitutes an actual cancer cell to be called cancerous. And then, like I said, at the very end here, we'll look at a little bit at prokaryotic cells. And we have to, it's some similar aspects, but some differences compared to a eukaryotic organism. But we need to talk a little bit about a prokaryote because we're going to manipulate those suckers. Because we can manipulate bacteria, we can manipulate a virus, and we can now inject DNA that we want to inject into other things, like plants, like corn, like us. That's how we do it with vaccines. Introduce a vector. Usually it's a bacteria. And so we got to look at a prokaryotic structure of genetic material as well. So we'll look at that. And it's pretty easy. It's pretty basic. It's, as you would expect, easy to manipulate if we can willy-nilly manipulate it all the time. The cell cycle, guys, this is where we're going to start our whole journey here, this introduction to the cell cycle. The cell cycle is a orderly set of stages. What does that mean? We have step, 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 step. We have a number of steps in which one cell can now become two cells. Okay. One cell becomes two cells. And with this one, it's identical. Okay. It includes a phase of quote unquote rest and in a phase of division. Now, we named it interphase because we thought the cell was actually doing nothing at the time. Better microscopes, better information. And we find that it's actually doing a whole hell of a lot of stuff, and we'll point those out here. The second major stage, or part, I guess, of the cell cycle is actually the phases of division, okay? Okay, and this is where the cell is actively dividing, okay? Cell is actively dividing. And this is the cytoplasm dividing. Okay. So this is our cell cycle. So what does that look like? Excuse me. We have what's called interphase, and you can see the vast majority of a cell's time is spent in this phase of quote unquote rest. It's actually prepping for the next phase of division though. But as you can see, the vast majority of a cell's time is spent here, roughly like 90% or some nonsense. Then we have the phases of division right here. Okay, we have our phases of mitosis, which we're going to be looking at. Prophase, there's prometaphase, whatever, metaphase, anaphase, and telophase. And then we have our division of cytoplasm, cytokinesis. All right. So this is what we're going to be looking at. All right. The cell cycle includes interphase and then all of the phases of mitosis. Mitosis is just the phases of division: prophase, metaphase, anaphase, telophase, followed by cytokinesis. Yep, I said it fast, but you'll be able to say it fast soon. So let's start at the very beginning. Interphase. Okay, as you can kind of see, most of the cell's time is spent in this phase. 90% of the cell's time is spent here. I don't know if that's worthy of remembering, but it's pretty cool information. Okay? To impress somebody at a party, good God, I mean, you know, you'd be able to hook up with anybody when you give them that information, all right? When a cell is an interphase, it still does its normal stuff, okay? Still transporting proteins around, still doing stuff. It is just getting ready, though, just in case. Most cells, just FYI, have a shelf life, okay? They only last maybe, you know, a few days, like some of the cells within the digestive tract, or like red blood cells, they last around 120 days, all right? So every cell kind of has a natural kind of, you know, best by date. And so cells have to be ready to divide when cells become non-functional, we kill them and we need to make more. So cells need to be ready to divide and give us more cells, okay? So that's what this phase of interphase is, all right? They're still doing their normal stuff, but, you know, they need to be ready to divide at a moment's notice. There are a couple examples, and the picture back here shows it, okay? This kind of G0 phase. That's what that sub-zero means, not, as in it's not going to happen. Some cells only stay in this quote-unquote phase of rest, which means they don't divide. And nerves, muscle cells are really weird. Nerve cells are really weird in the sense that they don't divide. You get what you get, and that's it. Whatever mom makes in utero, that's what you get. And you whittle them away over the course of your lifetime. Now, there's been some new cool research to suggest that actually nerve cells can regrow. So we're talking about the nerves in the brain actually regrowing, all right? Because we've thought for many years that they can't at all. And in the region where we can form long-term memories, the hippocampus, parahippocampal region, we've actually seen some cells or some neurons actually dividing, very small amount. So there is some dividability within the neurons, but for all neurons in general, that you basically have what you have and then you whittle them away, okay? You could whittle them away with disuse, not stimulating the brain. You could whittle them away with taking, you know, drugs, recreational drugs. Hell, probably some medications can affect brains, and some of them do. But whether or not it destroys nerves, I couldn't attest to that off the top of my head. But everything, diet, okay, everything that you are putting into you was going to affect the health of these nerves, all right? But I'll get back on track here. All right, this is kind of bonus type stuff right there, okay? But interphase itself, this quote-unquote phase of rest, actually consists of 3 different phases, okay? Just like the part of mitosis has prophase, metaphase, anaphase, telophase. Interphase also has some subphases associated with it, and we call them G1, S, and G2. What do these really stand for? Well, easy peasy. G stands for growth, and S stands for synthesis. T-H-E-S-I-S. Okay. Sometimes this is referred to as gap as well. So, but it's still a G. I call it growth because that's what they used to call it way back when, back in the dark ages. I believe now the textbooks call it a gap 1 and a gap 2 phase. But growth is just fine. And if you can use either one, but I'm going to try to use growth, but gap is the technical new name, but growth is, you're going to know it just perfectly fine that way. So when we look at these three, subphases, if you will, of the interphase, there is a specific thing or a specific couple things that are happening in each one. If I was a betting man and I was trying to figure out what in the world Dr. Thornley's going to ask on the exam for a potential short answer question, I would probably put all my chips in the middle on asking the cell cycle. I would know that interphase consists of a G1, an S, and a G2 subphase. I would know what happens in each of those phases. So in G1, basically all my organelles are doubling up. One cell is becoming 2 cells. So would it stand to reason that both of them will basically need mitochondria, smoothie R's, rough R's, all of those organelles that we talked about in the beginning? Of course so. All right. So as the cell starts to double up into organelles, would it make sense that it would grow in size a little bit? Right? More stuff inside means it gets a little bit bigger. This is the major thing that happens here. Okay. Organelles are doubling up. Organelles are doubling up. In S phase, we are synthesizing genetic material. What the hell does that mean? Well, this is what it means. We are making a copy of the DNA. The DNA of one cell needs to be copied so I can put it into the new cell. So this just means we are copying the genetic material, okay? And that's synthesis. Okay. This is kind of what it looks like. Okay, this jumbled mess. This is inside the nucleus. This is the nucleus, and this is the cell membrane, right? So here, all of this genetic material is doubling up. If we look at it close up with some electron microscope action and whatnot, you can kind of see that chromosomes enter with this thing called one chromatid. Like, okay, I don't know what the hell's going on. Don't worry, we will. And as it doubles up, there'll be another one associated with it. And it'll double up and it'll start associating with another one. In this phase, it is because there's so much genetic information inside the nucleus, it is very difficult to see all of these individual chromatids. So from a microscopic standpoint, using a microscope, you will not see the individual chromatids, but that is, in a sense, what is happening here. All you do is you see this jumbled mess, okay? You see a jumbled mess. Again, these are really super small. Seeing something at 40x, you ain't gonna see jack squat. G2, what happens here? Well, usually everybody just says this is the catch-all. Whatever didn't get synthesized in G1 gets synthesized here in G2. So if there's any organelles that didn't get duplicated, that happens here. But really, the major thing that happens is we synthesize the proteins. You're like, man, what? Why do we need to synthesize proteins? Proteins do a number of fantastic things, right? We've talked about that over the course of, it seems like the entire semester, but in chapter 5, we really talked about that, right? With how they are embedded in membranes and allow stuff to come across and help to move things across. I think in a typical cell, guys, there are 42 million proteins. 42 million proteins. That's like insane. So proteins do a lot of stuff. They help make structure. They help make shape. They help move things around. They help bring things across the plasma membrane. So synthesizing these proteins is going to be a very necessary evil. So I have the first phase of the cell cycle, interphase, G1, S, G2. G1, organelles double up. S, DNA doubles up. G2, any organelle that didn't get doubled up, it occurs here. Proteins get synthesized. This is interphase. And every cell, except for nerves and muscles are a little weird as well, go through this cycle completely. G1, S, and G2.
The replication of DNA and...
I need to... Let me increase my... sound real quick, guys. All right, let's try that. Share again. Sorry about that.
The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis.
Okay, so that's where we're at. We're prepping for the divide. G1, S, G2. Organelles double up. DNA doubles up. Proteins, enzymes double up. Now we're going to go through these phases of division. But before we get to these phases of division, okay, oops. Let me get my pen back. Before we get to these phases of division, either nuclear division or division of the cytoplasm, division of the cell, division of the nucleus, division of the genetic material, or division of the cytoplasm, we can actually control this. And by we, I mean our bodies. Okay? We can control when we enter into this thing called the phase of mitosis. We have a number of checkpoints. 2 checkpoints within interphase alone that ensures that our genetically identical daughter cells, and when one cell divides, it gets me two identical daughter cells. Okay. In order to make sure that they are genetically identical, because I want skin cells to become skin cells. I want liver cells to be liver cells. I don't need liver cells to be uterine cells. It ain't going to do a damn bit of good up there in the liver. Okay. So I need these things to be genetically identical. I have a series of signals that get released at certain points during this cell cycle. And when those signals are received, the cell goes, okay, here's my signal. May I continue? And we go, yes. And if for some reason that signal is at the wrong time or that signal is different, then the cell says, stop. Come here. Let's fix you up. Okay. And it'll fix the defect within the genes, which codes for the signals. And therefore, we can prevent a non-functional cell from continuing on, or another way to put it, a cancerous cell from continuing on. So we have a number of signals that the cell will release. We have growth factors. Okay. These are on the plasma membrane, and they signal that, hey, we're growing. Is it cool? Yay or nay? We also have this kind of family of I'm just using a very general term on the inside, cyclins. And these are just a natural number of proteins that are inside of a cell that increase and decrease, that kind of wax and wane, if you will, during the phases of interphase, G1, S, and G2. And it's these internal ones that really allow the cell to be governed, okay? Because if these cyclins aren't exactly correct at the right specific time, then hopefully the cell gets stopped. They call it arrested, okay? It just halts, okay? And this allows the cell, this allows the body to repair that defect. Because if something's wrong with that cyclin, chances are that there's something wrong with the genetic material inside that cell. There's a defect. And we don't want to copy a defect, okay? At least for this purpose, we don't want that happening, okay? Because if we copy that defect into the next generation, then the chances of that next generation of cells functioning properly goes down. And if that defective DNA is allowed to copy, well then from there on, I'm just going to have a defective functioning structure. And I don't want that. Damage by solar radiation or X-rays. When you go to a doctor's office or a dentist office, right? You get exposed to X-rays. That's why they drape you at the dentist office in so much lead aprons, because that exposure to those X-rays can cause damage to the DNA. That's why you wear a lead kind of metal guard over your gonads so you don't blow up your X-rays. Okay.
During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all 3 phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur.
And this concept of nutrition is one of the most important ones that we have. Understanding how nutrition can affect the genome and how genome can affect how we handle nutrition, there is a ton of research into this now. And they've discovered that the presence of certain genes passed on by mom and dad, okay? So this is going to be influenced by mom and dad's genome and their diet. You can handle certain foods better than others, like carbohydrates. Having a natural birth through the vaginal canal, having been breastfed, you are seeding your large intestines with a better group of bacteria. And that bacteria is better able to handle carbohydrates than people who weren't vaginally birthed and were not breastfed. And I believe there's a study to come out, I think next year, looking at individual parts of this process. Because if you had both, then your microbiome, the bacteria that live in your large intestine, are far superior than if you weren't breastfed or if you weren't birthed vaginally. So they're finding that a lot of processes go into this effect and affect the cell line that you have within the large intestine. So going in and making sure that your genome is optimal from mom and dad. Hopefully, you know, dad ate halfway decent and mom is eating halfway decent, okay? And they didn't get exposed to some kind of cancer-causing agent, a teratogen, that ensures that all of these checkpoints are perfectly cool and kosher. And if they are not kosher, normal function cell can stop and fix it. If it's not kosher and it doesn't get stopped, that's when we have potential defects in the genome, in how the cell divides, okay? Because we have this thing called Down syndrome, right? It's trisomy 21. It's because chromosomes didn't divide equally. Well, again, that goes to this checkpoint right here that they just mentioned, okay? There wasn't a proper division that occurred. And sometimes the DNA becomes defective as it gets older. So our ability to see that, stop it, fix it up, or kill the cell doesn't properly happen. So, you know, age of a cell, size of a cell, the nutrition associated with cells, all of these go hand in hand in helping to prevent abnormalcy to occur.
During the cell
This ability to stop that during the cell cycle and try to fix it is awesome. But if we can't, we've got to be able to kill this thing. And that's what apoptosis is. Apoptosis is programmed cell death. Okay? And this involves a number of processes. I'm not going to ask you all the individual steps. It causes some blistering, basically, of the plasma membrane. It'll cause the fragmenting of the nuclear membrane as well. Causes these things called bleebs to form, and basically makes it really easy for this cell to be self-destructed. And this is kind of what it, this is kind of what it looks like where it just kind of starts to break apart. And then we can get, this isn't normal. So we have other cells in the body that see, oh my God, that's not normal. Let's go over and kill it. So they'll go over and chew it up. And it's basically different types of white blood cells that go over there and see it and destroy it. So that is what apoptosis is. All right. Apoptosis is fantastic. Okay. It's why we have 5 fingers. In utero, we have webbing in between our fingers and toes. And apoptosis gets rid of that skin in between our fingers. Okay. So now I have 5 fingers instead of a webbed type hand. That'd be awesome at swimming. Okay. But, interacting with the world, that wouldn't be probably the case. They are opposing forces. Yes. Mitosis looks at to increase cell numbers. Apoptosis looks to decrease cell numbers. Okay. Yeah, I guess. I mean, the book makes a big deal about it, but cell death means their numbers are going to go down. So it's just intuitive that they're opposite opposing forces. But apoptosis is perfectly cool. It's perfectly fine. All right. This happens all the time. If a cell's not functioning, what do? We kill it. We don't need it. I won't ask you the P53, so don't worry about that. I won't ask you the specifics, signaling protein involved with this. But again, it's just there if anything is abnormal. Okay. Now, cancer cells, they can screw with this apoptosis. Okay. They can manipulate that cyclin, that signaling protein, to be just a little different. And it's just that little bitty difference that can cause a cell to become cancerous, where I can't control the cell cycle. And I allow the cell to divide and divide and divide and divide. And when it comes to cancer, I don't want that. Okay, you don't want that. But if a normal acting cell does become weird and it's precancerous, I can get that thing. No worries. I can get that thing to die. Okay. And all of these enzymes, all these signaling proteins, they're inside each individual cell, guys. They're inside each individual cell. There will be promoters and there will be inhibitors of it. Okay. If the cell is perfectly fine, there'll be an inhibitor and the cell continues. All right. If there's some kind of defect, well, then we'll promote the destruction of this thing. And it all has to deal with proteins, which is why there's like 42 million of them in each individual cell. Okay.